Okay, now let's get started. Welcome everyone to the RNA Collaborative Seminar Series, sponsored by RNA Society. So our RNA Collaborative Seminar Series include 33 centers all over the world. So during the seminar, please do not take video or photos, because we are going to, all the talk will be uploaded to the YouTube channel, so you can watch it anytime. So you just need to subscribe it. On today's seminar series will be, I have two centers. One is the Zhejiang University Center for Medicine. The other is the RNA Institute at the University at Albany. So I'm the center director for the Zhejiang University for Center for RNA Medicine, Xin Li. So it is my great pleasure to introduce our first speaker, Guangnan Li, a postdoc fellow at our center. So Guangnan received his bachelor degree in biopharmacy at Wuhan University, and he continued his PhD training at Wuhan University, major in biochemistry, and mentored by both Professor Xiangdong Fu and Professor Yu Zhou, where he developed a strong, solid training on RNA biology. And in our lab, so Guangnan has pushed this experience into germ cell RNA biology and piRNA biogenesis. Today, he will tell us about a new function of ribosomes that is a function during the piRNA biogenesis. Without further ado, join me to welcome Guangnan Li.
Okay. Okay, thanks for Professor Li's introduction. Hello, everyone, and I'm a postdoc from Center for RNA Medicine, Zhejiang University. I will tell a story about how ribosomes do more than just translate. And it's a story about how germ cells rewire translation termination to produce pines. Okay. The pines are small RNAs quite different from microRNAs. So they are longer and bound to PV protein that have 3'O methylation modifications. And most of pines are expressed in germline cells. Another huge difference from microRNA is that pine has much larger species, nearly 500 times microRNA species. You can see here. So this is due to the unique biogenesis pathway of pines. So not like microRNAs that are processed from stem loop structures that have paired double-strand RNAs by endonuclease of DOTA and digital. The pines are manipulated from single-stranded RNAs with M7G cape and polyA tail, and cleaved by PLD6, which is an endonuclease highly expressed in germline cells. And PLD6 is also a membrane protein that's anchored to mitochondrial outer membrane. So as early as the year of 2020, our group have found that pine precursors can be translated and produce small peptides. So furthermore, our group have also found that after translation finish, the ribosomes don't leave and go on into the supreme UTR. And more detailed study indicated that 5' ends of the ribosome protective fragments share the same sites with pine 5' ends. So that study also leaves a scientific question unsolved. It is after finished translation, why don't ribosomes simply terminate, and why they don't leave? So for normal termination, F1, F3A hexadimer plays a crucial role. The YAP1 mimics TR structurally to recognize the stop codon, and YAP3A hydrolyzes GDP to regulate the conformation change of YAP1. And the two factors initiate the recycling of ribosomes after translation termination. Recently, a new technique is created to detect the YAP1 recruitment at the stop codon, named YAP1-seq. So it uses YAP1 specific antibody to pull down YAP1 ribosome complex, then use the RNAs from the YAP1 ribosome complex to construct the Ribosome library. So this technique shows a very good performance on representing YAP1's enrichment at stop codons. To answer the scientific question that during piRNA processing, why don't ribosomes terminate and leave? We apply YAP1-seq in mouse tested samples. We find that YAP1-seq shows good performance on mouse tests, and an obvious peak at stop codon can be observed on canonical MRIs. But when we turn to pine precursors, we only see a mild peak at stop codon. This means that YAP1 binds less efficiently to stop codons of pine precursors. Then why does YAP1 bind less efficiently to stop codon? Actually, as early as 2020, our group has noticed one factor show effects on ribosome occupancy on circulating UTR. And this factor is MALT1 protein. So when we do conditionally knockout of MALT1 on adult mouse tests, we see elimination of pectin piRNAs. We also see a dramatically decrease of ribosome occupancy on circulating UTR in MALT1 CQ samples. You can see here. So this phenomenon lets us to focus on MALT1 protein. We do YAP1-seq in adult mouse tests that have MALT1 being conditionally knocked out, and find that compared to control group, loss of MALT1 enhances the YAP1 recruitment at stop codon region, specifically on the pine precursors, not the control mRNA. I also show a check picture here on the right, and you can see an increased peak, specifically at stop codon in MALT1 secure group. The MALT1 is a 5' to 3' RNA helical that has already been reported, but we don't have good antibody to study this protein. So we constructed a mouse tool. So this mouse has a flat peg inside the MALT1 coding region, and this mouse shows normal fertility and testis morphology. So we capture MALT1 interacting protein using this mouse tool and identify them by mass spectrometry. From the result, we see MALT1 can pull down known pine factors like TV1 and TV2. Furthermore, we see YAP3 is included in MALT1 interaction. And we further validate MALT1 YAP3 interaction in HeLa cells by ectopically expressing these two proteins. So we also divide YAP3A into 4 domain parts and check which domain of YAP3A interacts with MALT1. And from this result, we see C-terminal domain of YAP3A interact with MALT1 and with the same binding ability with the full-length YAP3A. Yes. So it is worth noting that the C-terminal domain of YAP3A is also reported to interact with YAP1. So we query that whether YAP1 can interact with MALT1. So we use YAP1 specific antibody to do IP and find YAP1 can pull down MALT1 in an iron-independent manner. So in vivo, If we want to get obvious YAP1, YAP3 artifacts, you need to use GTP analog to stabilize YAP1, YAP3 dimer. Here I use GPT-CP to stabilize YAP1 and YAP3A. You can see that without this drug as stabilizer, the YAP1 binds to less YAP3A, and it also binds less molten L1. So this indicates that molten L1, YAP3A, YAP1 can form a dynamic complex. So combine all these results above, we find the molten L1 shows ability to attenuate canonic termination by engaging YAP1, YAP3 interface, and this may induce the ribosome bypass stop codon and pine precursor. Then we meet a new problem. How to ribosome translocate beyond stop codons? There are two models. So one has the 80S disassembled into only 40S binding to RNA. Then the 40S sliding into 3' nuclei. Another has intact 80S keep sliding into 3' nuclei. So we further do a 40S footprint to check whether the 40S sliding exists on pine precursor. The 40S footprints show an obvious enrichment of signals at 5' nuclei and subcodon region on canonical mRNAs. When we focus on pine precursors, we see 40S spreads into the 3' nuclei region. You can see a bunch of signals exist here. So there comes a question that where does this 40S ribosome come from? So there are three possible models. The first study is this 40S come from internal initiation. The second 40S come from leaky scanning from the very far term end of the mRNAs. The third, the 40S come from the unrecycled ribosome, which means the termination blocks the ribosome is disassembled into 40S, and 40S further again the 3' nuclei region. We do 40S footprint after inject heritonium into mouse testes, and to inspect this model, these three models, we plan to use heritonium treatment, because heritonium specifically blocks 80S elevation. So only the third one, unrecycled 40S model, is sensitive to heritonium treatment. So we do 40S footprint after injecting heritonium into mouse testes and find different UTR 40S ribosomes are sensitive to heritonium treatment. So this leads us to the unrecycled 40S model. That different UTR 40S ribosome comes from the unrecycled 40S. The further analysis of ribosome footprint data show that 80S RPF share overlaps the 5' end with piRNAs, and a similar phenomenon is also observed from the 40S RPF data. So this indicates that both 40S and 80S ribosomes participate in pine processing. Then, which type of ribosome is involved in cleavage? Is it 40S or 80S? It is well known that 5' and pines are determined by PLD6. So PLD6 is a mitochondrial autonomous protein and locates two intermitochondrial segments impacting spermatocytes. It forms dimer and shows the 5' U cleavage preference in vivo. Then the question changed to which subunits, 40S or 80S, participates in PLD6 mediated the cleavage. So we collect the 40S and 80S, then do PLD6 immunoprecipitation separately for detecting footprints from PLD6 ribosome complex. And the results show that only in 80S fraction, PLD6 can pull down the iron fragments from pine precursor supranuclei. These supranuclei are pine-derived regions. So until now, the evidence indicates that a model that pectin pine precursors 48 ribosomes translocate to supranuclei, but 88 ribosomes participated in the PRD6 cleavage. To decipher how PRD6 interact with ribosomes, we also create a mouse tool. This mouse has a black tag on PRD6 C-terminals. So this mouse also shows normal fertility and testis morphology. So after using this mouse to do RT-mass spec to identify PRD6 interacting protein, we find PRD6 not only binds to known piRNA biogenesis factors like including the Myotonic or P2, but also binds to one large subunit protein, which is RPL34. So in ribosome crystal structure, the RPL34 locates near the E site of 88 ribosome. We next do a propeller assay to validate the PRD6-88 ribosome interaction. This assay incubates the purified PRD6 protein with different subunits of ribosomes, then do an ultra-speed spin down to collect pellets for Western blotting. From these results, we do see PRD6 copellate with the 68s and 88s. So until now, we have a more clear model. So after Myotonic block translation termination, 48 ribosomes run into supranuclear. Then we assemble APAs to form intact. Then we assemble 68 to form intact ADAs. And PRD6 will interact with E site with ADAs ribosome to cleave iron. However, the PRD6 alone shows no sequence preference in vitro. But it is well known that pine have a 5' U preference on sequence. So how is PRD6 cleavage regulated in vivo to generate a 5' U bias? So when we do PRD6 ecliptic from adult mice testes, we see PRD6 specifically bind to pine precursors. When we do PRD6 ecliptic under the Myotonic CQ background, we see PRD6 binding to pine precursors is compromised in the absence of Myotonic. So for ecliptic data, we can use CITS to call the direct protein iron cross-link sites. So for PRD6 CITS, clipped CITS, we do an analysis to show the distance between all CITS position and pine 5' N. We see peaks that indicate PRD6 bind to future pine cleavage sites. That means PRD6 in vivo can recognize future pine cleavage sites. But the loss of Myotonic will eliminate this peak. That means Myotonic can help PRD6 to recognize future pine cleavage sites. Then how does Myotonic facilitate the PRD6 recognition of pine cleavage sites? So from Myotonic ecliptic, we see Myotonic also specifically binds to pine precursors. So from meta-analysis, we see Myotonic strongly binds to pine precursors, and its binding is throughout the entire RNA. We also apply CITS analysis on Myotonic eclipse data and find Myotonic binds to a new iron motif, which is a U-rich motif. This motif matches 5' U bias of piRNAs. The further analysis indicates CITS lies immediate upstream of ADS and pine cleavage site. This means Myotonic can recognize future pi activity sites in vivo. So from the previous IP mass spec data, we have already seen the MOLT1 36 can pull down each other. So we further validate this interaction after expressing MOLT1 36 by doing flag IP in HeLa cell. We also collect 40S and 80S fractions separately for MOLT1 immunoprecipitation to detect footprints from MOLT1 ribosome complex. This result indicates that MOLT1 pulls down iron fragments from pi-derived hotspot region only in the 80S fraction. So this indicates that MOLT1 interacts with 80S ribosome at time precursor supranuclear. Now we are clear that MOLT1 36 can form complex to bind near ribosome E site. Since MOLT1 is able to recognize the U-rich motif, can it independently recognize pi activity site in the absence of p86? So from the protein distribution results, we see MOLT1 not only to sediment with the mitochondrial fraction, but also remains a lot on 3 RNA fractions. So we collect these two fractions and do MOLT1 ecliptic separately. So from the CITS analysis, we see in 3 RNA fraction, MOLT1 shows very low ability to bind at future pi activity sites. This means that MOLT1 may be not able to recognize pi activity sites alone. Then we query that whether PLD6 will promote MOLT1 binding. So after analyzing MOLT1 ecliptic data under the PLD6 conditionally knockout background, we see that in the absence of PLD6, the MOLT1's ability to bind to future pi activity site decreases a lot. So all these results indicate that MOLT1 PLD6 coupling promotes the efficient recognition of future pi activity sites. So finally, combining all these evidences, we propose a more comprehensive model to explain how stem cell Rewire translation termination for pi biogenesis. So pectin pi precursors possess the canonical 5' caps and polyA tails, and in cytosol, they can be translated into small peptides. However, MOLT1 specifically binds to these RNAs and interfere with translation termination by sequestering the F1 and the F3 complex. This causes the disassembled 48 ribosome subunits to slide into the supranuclear. Then in the mitochondrial cement, which is a specialized subcellular compartment enriched in piRNA-specific factors and precursors, 36 and MOLT1 together with the 60S subunit rejoin the 48 subunit to form a complex. So this complex recognizes the U-rich sequences to promote 36 mediated cleavage, then generates a 5' U containing iron fragments that are subsequently matured into pines. So this concludes all my presentation. So finally, I sincerely thank my mentor, Dr. Li, for his continuous guidance, and thank all the collaborators for their essential contributions. And I welcome any questions and suggestions.
Okay. So if you have any questions, you can type in the Q&A section, so I can read it to Guangnan. So Guangnan, would you mind to maybe give a little more background on how the MOLT1, what is the current evidence how the MOLT1 bind to the specifically bind to the piRNA precursor?
The motor one, it is a known helicase. It has a helicase R helicase domain on its C terminals. So the helicase domain is a canonical domain that combined the two RNA. From our data, we see the motor can specifically bind only bind to the pectin pi precursors. I think that they may have other time factors to help it because in because normally we think that the helicase has no strong specificity on R sequences. But we do find the multi-L1 can recognize the U-rich motif. And in this story, we found that the PLD6 will have a multi-L1 to recognize that motif. So in the germ cell, I think multi-L1 alone might not be enough to especially bind to this precursors. And from our method data, we do find the PV1, PV2, and the TGRD1, TGRD5, and other known pyogenic factors. So I think they may form maybe in such of before the multi-1 enter into the intermitochondrial cement, and it already forms some complex with other factors, and this complex can specifically recognize and bind the prime precursor.
Okay.
Then yes.
Okay, I think it's good. Good. So now there's a question pop up in the Q&A. For people who want to ask a question, also if in the panel, so if you want to ask, you can just raise a hand, so you can use the mic, and you can type in the question. So first one is, can this molten L1 PLD6 complex with ATS ribosome and piRNA be reconstituted in vitro?
We are trying. We have purified the PLD6 and we do the co-pellicle assay, you see. We get the information that the PLD6 and the ATS can be co-pellicated, but the single particle structure, they cannot get the single particle structure. So then that means that the PLD6 along with ATS is not stable. We now also find a method to purify multi-L1 now, but it is still ongoing that we haven't get a large abundance of this multi-L1 protein now. So after we get the protein, we will try to see whether this three can form a stable complex. Yeah.
Good. So the next question is that are the mRNAs from which the piRNA are made merely upregulated when piRNA processing is perturbed? I think they're asking about the piRNA precursors, whether they are upregulated when the piRNA processing is perturbed. And are different mRNA affected by knockout of different processing factors?
Yes, among these processing factors, now we only know that only multi-L1, PLD6 knockout can increase the prime precursor mRNA a lot. And because these two factors are known the most upstream factors. So there are also other factors, but like the TDRD5 protein or other like what we know that PV protein. But when you knock out this protein, the R cleavage will still happen. So because of the cleavage, the stability of this prime precursor is not good. So that won't increase the R a lot.
So basically, it is upregulated in the mostly packaged
Yeah.
piRNA, right?
Yeah.
Okay, I think we can now time it. Is up, so we like to thanks Guangnan for this seminar, and thanks for people asking the questions. And now I'm pass the torch to Andy, the Albany Center at Albany. Go ahead.
Great, thank you. Great to be here. I'm Andy Berglund, Director of the RNA Institute. It's my pleasure to introduce Dr. Cécilia Légaré, postdoctoral fellow in my lab and jointly mentored by my colleague Elise Duchesne at the University of Laval in Quebec, Canada. And Cécilia, take it away.
So thank you for the introduction. So today I'll be presenting part of my postdoc project and looking at alternative splicing changes over time in individual with the spliceopathy disease, myotonic dystrophy. So I'll start with a brief overview of myotonic dystrophy type 1 or DM1. So it is caused by a CTG repeat expansion in the 3' untranslated region of the dystrophia myotonica protein kinase gene. And what happens is that the RNA that contains the CUG repeats form these hairpin structures, trapping both splicing factors and transcription factors. So that affects both messenger RNA expression, microRNA expression, as well as alternative splicing. The global prevalence of DM1 is 1 in 8,000. However, in some regions it's much higher. In the Saguenay-Lac-Saint-Jean region, from where I'm from, it's 1 in 630. So that's in Canada, Quebec. So as you can see here, by the different individual, DM1 is highly heterogeneous disease. And if we go into more detail into the clinical manifestation, you can see here that it affects almost all systems. Here are just a few examples. So for example, the endocrine system is affected, the cardiovascular system, the ocular system, the cognitive system, the respiratory system, and the gastrointestinal system. It is a dystrophy, so the musculoskeletal system is also affected. One of the major symptoms is myotonia. For those who don't know what myotonia is, it's an inability to release muscle after a contraction. So you can see here the patient squeeze his hands and it's taking him a very long time to open his fist. So that's the symptom which is called myotonia. There is also some muscle atrophy and weakness. And I mentioned it was a highly heterogeneous disease. It has multiple different classification or phenotypes, mostly based on age at onset, but also on CTG repeat length, the earlier phenotypes having more longer repeats than the one with later age at onset. So I'm presenting here the 5 phenotypes. So the congenital, the symptoms would appear in less than 1 month, the infantile between 1 month and 10 years, the juvenile between 10 to 20 years, the adult 20 to 40, and then the late onset later than 40 years. I'll be coming back to the phenotypes later in my presentation. So there have been some clinical studies looking at the changes, clinical changes over time. This is one of these studies that has been made by Xin Zhiguo Li in our group. And you can see that there is some muscle strength changes in the M1 over a 9-year period. So you can see here a strength decline over 9 years for all muscle groups except for X flexor, with some of the more distal muscle like wrist extensor here and ankle dorsiflexion showing the greatest decrease over a 9-year period. They also did a follow-up study with a shorter time frame. So they look at muscle strength changes in DM1 over 3 years. You can see that there is less muscle affected, but you still see, so those that have box in red, so there is a reduction in grip strength, lateral pinch strength, hip adductors, knee extension, and ankle dorsiflexion. This time, both knee extensor and ankle dorsiflexion loss was similar. So there is a significant loss of grip, lateral pinch, hip abductors, knee extensor, and ankle dorsiflexor strength over a 3 years period. So I'll mention that alternative splicing was dysregulated in DM1. I'll just make a quick reminder of what alternative splicing is. So you have constitutive splicing here displayed, where the introns are removed and then the exons are stitched back together to form the mature messenger RNA. Alternative splicing is just another way. So here you have the skip exon events where one of the exons is skipped, so it's not included in the final isoform. And why this is important, it's because there are some examples of missplicing in DM1, and some of them are related to some of the clinical manifestation directly. So here you can see in an affected individual, MBNL will repress the inclusion of exon 7A in the CLCN1 gene, which will then create a normal messenger RNA, a normal protein, and then a functional CLCN1 channel. However, in DM1, MBNL1 is trapped in the nucleus, so it cannot repress the inclusion of this exon 7A. So in DM1 patient, this exon 7A is included and is then degraded by nonsense mediated decay, leading to a reduction in chloride ion conductance and the myotonia symptoms that I showed. To measure the alternative splicing, we're using the percent spliced in, or PSI, for which we take the longer isoform over the total, and we get a percent. So this is how we measure alternative splicing. So this is how to measure it for one event. However, as I mentioned, there is multiple events that are dysregulated in DM1. So to have a better understanding of how dysregulated the splicing is, people have created splicing index score to have a better idea. So this is from a study made by Provenzano and al., where they make this splicing index score, also known as a composite alternative splicing index, or the CASI. You usually extract RNA from human biospecimen. In this paper, they used the tibialis anterior muscle. And then you can run targeted RNA sequencing of these 22 skip exon alternative splicing events that are known to be dysregulated in DM1. You get PSI values for each of the events, and then you do a normalized PSI calculation. In order to do this calculation, you need to have both median PSI control values. So you get these from an external cohort, as well as a 95th percentile DM1 value. So they made an external cohort and get those variables. So then you can take your normalized PSI values for each of your events, remove the median PSI value and then of the control, and then divide that by the PSI of the 95th percentile DM1 minus the median of the control, and then you get your normalized PSI for that particular event. To compute the SI score, you average these normalized size for all 22 events. And Provenzano and all actually divided in 3 different groups the SI score. So the mild group, which has an SI score between 0 and 0.4, the moderate group, which has an SI score between 0.4 and 0.75, and then the severe, which is between 0.75 and 1. What is interesting with the SI score is that it also correlates with clinical performance. So here is clinical performance compared to reference values, where you have the severely affected being at 0 and the unaffected being at 100%. And as you can see, patients with more severe SI score also perform more poorly on clinical performance. So here are some results that I'm showing from the study from Provenzano and al., where you can see that the SI score, which is displayed here, is actually correlated with the percent predicted strength of ankle dorsiflexion, with patient having the more dysregulated SI score here, also having the less strength in percent predicted. There is a similar correlation with NGRIP, as well as the 10-meter run fast walk test, which is a test where patients are asked to walk or as fast as they can or run on a 10-meter distance, and then you calculate how long it takes them to do the test. Again, the higher their splicing index score is, and the longer it takes them to do to walk or to run to do that distance. In this study, they also look at changes over 3 months of time. So they had biopsies for a subset of patients which were taken 3 months apart. And you can see here that there is a slight increase, however, not significant, of the splice index in the mild group, a significant increase in the moderate group, but almost no changes in the severe groups. So currently we know that the SI score correlates with clinical measure and progress over 3 months in TA, which makes it a good biomarker. However, the vastus lateralis muscle is frequently simple in clinical trials and is also important for daily function in the M1 patients. It is a muscle that is important if you want to get up from a chair, if you go up and down stairs. And little is known about splicing in this muscle. And we also don't know how splicing progress over a longer period of time. So this is where my study comes from. So the objective of my project was to evaluate the association between clinical measure of muscle strength and function with SI score derived from vastus lateralis biopsies. We also wanted to assess changes in SI score over a 3-year period. So in order to do so, I had access to 2 different cohorts. So a cross-sectional cohort of 51 DM1 patients, for which I had vastus lateralis biopsies, and we used targeted RNA-seq to generate the SI score. We also had for these patients lower limb quantitative muscle strength expressed as percent predicted and grip strength also expressed as percent predicted. For the second objective, I had access to, in a subset of patients, to biopsies taken 3 years apart, in a subset of 20 patients. And you can see here that we also used the same vastus lateralis biopsies and targeted RNA-seq, and we also had the same clinical measures. So here are the baseline characteristics of the cross-sectional cohort, which has on average 47 years of age, where slightly more females and more juvenile and phenotypes. And you can see that on average, the patient had, so this is the strength of knee extensor, ankle dorsiflexion, and grip strength as expressed as percent predicted. And you can see that they are lower than what is expected. And the splicing index score average was 0.3. So the first thing we did was to see if the correlation between muscle strength and SI score also holded true in the vastus lateralis muscle. And you can see here the normalized SI score as well as ankle and percent expressed as percent predicted. And you can see that for a participant who has a low SI score, they had a high ankle dorsiflexion strength, whereas a patient with high SI score has low percent predicted for ankle dorsiflexion. There is a similar results with knee extensor here and grips. So we were able to validate that the SI score correlate with muscle strength in vastus lateralis, which is consistent with data in the TA muscle. We then wanted to see if our cross-sectional and longitudinal cohort had baseline were similar. So you can see here the comparison of our longitudinal and cross-sectional cohort. And you can see that they're very similar in both age, sex, phenotypes, and CTG repeat length. We then look at clinical and SI changes, and SI score changes over 3 years. So here you can see for the knee extensor at baseline, it went down from 51 to 47%. But this was not significant. Similarly, the ankle dorsiflexor strength went down from 55 to 49, but it's also not significant. The grip strength did reach a significant decrease, going from 46 to 41%, as well as the SI score showing a significant increase, going from 0.3 to 0.5 over that 3-year period. To have a better understanding of the changes over that 3 years period, we put our participants in the same tree grouping of SI score that was used by Provenzano and al. And this is a very busy figure, so I will spend some time to explain it. So the shape here represents the number of CTG repeat length, diamond being less than 300 CTG repeat length, circles being 300 to 800 CTG repeat length, and then square being more than 800 CTG repeats. The color here are for the phenotypes. So we have in pink here the adult phenotypes. We have some late onset here in gray, and we have most of the juvenile onset patients here in blue. So here you have, you can see that there is a significant increase in splicing index in the mild SI score group. And you can see that, however, you can see that it's very different from one patient to the other. You have here the late onset group that don't seem to change much. You have some participants here that have dramatic increase. Similarly, for the moderate SI score, you also see an increase. However, the severe SI score participants don't show any changes in SI score over that 3-year period. So this was similar to the results obtained by Provenzano and al., where they didn't see an increase in the severe SI score groups. They did see more difference in their moderate SI score group than in the mild. So this is a little bit different, but it is a different muscle and a different period of time. As the phenotype seems to have some effect, as well as the baseline splicing index, we wanted to see whether or not they had an effect. So we built regression models. So here you can see regression models for the changes in SI score, and we use both baseline SI score and late onset versus the other phenotypes. And you can see that they are both included significantly in the model and explain 31% of the variability of the changes in SI score. To have a better understanding of this association between baseline SI score and the changes in SI score, we plotted these two variables and did regression analysis. And you can see that the correlation is not really strong when you graph them. However, there is this group of participants that are late onset and also did not show any dysregulation at baseline. And when you remove them, the correlation now is significant and higher. With the less SI score you had at baseline, the more changes you have over time. And you also can see that these 3 participants seems to be very interesting because they change a lot. They all have, there's 2 different phenotypes here. So we have some adults and 1 juvenile onset, and they all have like different CTG repeat length. So in conclusion, the vastus lateralis's SI score is correlated with muscle strength. The SI score increased over 3 years, but the progression is variable. And it mirrors the clinical progression in cohorts. So this is also something we saw, that the progression was also variable when we look at the clinical variables. The baseline SI and phenotypes both partially explain the changes in SI score, and there seems to be a ceiling effect. So there's no rise in SI score with participants in the severe groups, which has baseline SI score over 75. The VLSI score is a good splicing biomarker source, but we need to have a better understanding of the variability of it. And like I mentioned, we had these 3 outliers that seems to be very interesting. So there's lots to learn from these 3 participants, which shown more dramatic changes in their SI score. So I would like to thank everybody in the Berglund lab, as well as the Duchenne lab and the Germ, the funding agencies, and the organizer for allowing me to present my results. Do you have any questions?
Great talk, Cecilia. I can start with a question to get the ball rolling. Do you, in terms of a comparison to the TA, you made some reference to similarities to the TA studies. I'm not sure you stated it. Do you think, is your impression that the TA missplicing would be more severe than the vastus lateralis? And why might you think that to be the case?
Yeah, so I'm not sure if I mentioned it to, or I think I mentioned it, that the DM1 is a more distal disease. So the more distal muscle are more affected. So if I go back, the TA muscle is its ear, so it's more lower than the vastus lateralis. So it, in theory, it should be more affected in terms of splicing as well, but we haven't. There haven't really been a study where you look at both vastus lateralis and tibialis anterior at the same time in the same patients.
Yeah, good point. You have a question from the audience. Ulrich asks, well, first, thanks you for a great talk, clear talk. He is wanting to know about the SI scores from the 3 outliers. And I think what he's asking is it possible those measurements are incorrect, or, you know, he's curious about the sort of how confident you are in the SI scores for the 3 outliers.
That's a good question. We did also, so didn't mention it, but we also did bulk RNA sequencing on these participants. And we do the SI score from the bulk and the targeted correlates very well. It could be, there are some people that are mentioning that because the biopsies aren't taking exactly at the same spot, you could have, you could hit like a more, like you could have a difference in terms of like the SI score from like one part of the biopsy, because the biopsy is not taking at the same space because there will be fibrosis. So, but we go like it's the same muscle and it's in the same region. They also, I don't have it on my slides, but they do correlate as well with the clinical. So we see some association between the clinical. So if the SI score progress, we also see progression in terms of clinical. We didn't see any clinical difference between these patients exactly and the rest of the cohort. So there's no like clear indication of why. I'm looking right now with the bulk RNA sequencing at gene expression changes. But I'm still in the initial analysis, so I don't really have anything to share for that.
I think you answered that question well. Well, there are no, does anyone, we have just a few minutes left if anyone has any questions before we wrap up. Sangeeta, anything to add as we wrap up this RNA collaborative?
Nothing else. I will update the speakers on the link to the YouTube later today.
Okay. Well, thank you all for joining.